Hello, my name is Derek Lawrence. This is my poster. The study was on porous fluorapatite and hydroxyapatite scaffolding grown with human osteoblast cells. These are bone grafts, and this is a pilot study. I worked under Dr. Jill Shea, Dr. Suji J. Polina, and Dr. Agarwal. This is a study focused on repairing bone injuries, specifically with these scaffolds, fluorapatite and hydroxyapatite. And we fabricated these scaffolds, compared them to each other, as well as a cell drop control, which is just the osteoblast cells grown in a 24 well plate by themselves and with media. And so the cells were also grown on the discs, and we predicted that the FA would show the most significant growth in comparison to the HA and the cell drop controls, and would also show signs of differentiation into bone cell lines that we could see the bone is growing and filling in the defect area. So we manufactured these discs, had the media ready for the cell culturing, and grew the cells on the discs as well as just on the plate itself for the cell drop control at specific densities that were essentially proportional. And then at 2-day and 10-day time points, performed an Almar Blue assay to determine cell viability, after which we fixed in 10% formalin, and then immunohistochemistry was performed later, primary staining for osteopontin and osteocalcin, as well as secondary staining to get the colors, and then immunofluorescent imaging was performed. And so what we saw was that on the day 2, there really wasn't much difference in growth between the mediums, the FA, the HA, or the cell drop control. And generally, there wasn't a ton of differentiation either. The cells were there and alive, but they weren't differentiating much. The HA may be showing slight differentiation, but little, if any. Maybe some in the FA as well, but little, if any. And then at the 10-day observed time point, there were significant differences in the HA, between the HA and the control, and maybe some with the FA, but essentially the control had the whole well to grow and continued growing and eventually started dying off because they overgrew. And the HA and FA slowed down, the HA more, with the HA and FA showing some differentiation, but the FA a significant amount more differentiation from what you can see in the IHC images for day 10, which shows that the osteopontin and osteocalcin were definitely showing on the FA, but not the cell drop and not as much on the HA. So the cells were growing, somewhat differentiating more on the FA, but growth was definitely slowing. And what we want to do is continue using this as an optimization for protocols as well as data analysis, and to eventually use these optimized results as ways to further medical science towards that of repairing these bone injuries in a way that can be beneficial to the public. Thank you.